agreement. Miss Soumya was asking one of the doubt called as what are microbodies? Here I wrote the definition. They are the minute single membrane bound organelles. Very minute, very small as compared to other organelles like mitochondria, chloroplast, endoplast reticulum, Golgi complex. These are very smaller. Also having a single membrane, and these are full of required enzymes for various processes in the cell. Okay, and there are two types in present microbodies, namely peroxisomes and glyoxisomes. Okay, these are two types of microbodies. They are also having a single membrane, and inside this we find different enzymes required for oxidation process. Here, one of the enzymes called as peroxidase is present inside the peroxisome. This one involved into the various processes of the plants and animal cells. Okay, and one more is glycosomes. These are certain enzymes required for the oxidation of fatty acids in seeds under germinating conditions. So there are also a kind of microbodies present in the germinating seeds of oil bearing seeds. Okay, so that are the microbodies. Very very important question you asked. The students asking a detailed study of the chromosomes once again. As we explained, various portions of the chromosomes here at the center, we find one of the constricted region called as primary constriction, also called as centromere, also called as centromere. So this primary constriction is a point where both arms of the chromosomes unite, or both arms of the chromosomes get attached to each other. It is a primary constriction or centromere. Okay, and these arms having two elongated lobes or structures called as chromatids. Chromatids are the lobes, and these chromatids having a coil-like structure inside it called as chromatin fibers or chromatins. So these chromatins are the actual sites of the chromosomes because these are formed by the complete DNA and histone proteins. Then at the certain regions, okay, not in all the chromosomes, in few chromosomes of the nucleus, we find unstaining structure called as secondary constriction. Secondary constriction. Secondary constriction is unstained here. Okay. After that, we find the remaining a small portion of chromosome, especially it is a heterochromatin, and that is called as satellite. It is called as satellite. The function of satellite is unique. It is used as a marker, uniquely identification of terminals through the DNA fingerprinting technique. Okay, and tip of the chromosome is telomere. It is a telomere. Okay, tip of the chromosome is called as telomeres. Okay, then one student asking satellite is unstaining. No, it is a secondary construction that is unstaining. Just above this secondary construction, we find a remaining portion of the chromosome, a special chromosome, special chromatid called as satellite. Yes, satellite is stained, but secondary construction is unstained. Okay, thank you.
One student asked a doubt about chromosome. You know very well that chromosome, it is a chemically made up of what? It is a chemically made up of nucleic acid. It is chemically made up of nucleic acid. So what type of the nucleic acid is present there? DNA. Okay. And along with this, even protein also. So only we are going to call as a nucleoprotein. Nucleoprotein. So here the function of this chromosome is it transfer. It is called as the hereditary vehicle. It is called as hereditary vehicle because it transmits the character from one to the another. So this is the main function of the chromosome. Is it clear? Because of presence of a genetic material called as the DNA. Is it clear? So in most of the organisms, DNA acts as genetic material. So it will transfer the character from one to the another. So that is the function of the chromosome.